Assalamualaikum and hello everyone. So today we are going to present the immunological aspects of SLE, which is systemic lupus erythematosus. So this is the members of our group. Systemic lupus erythematosus (SLE) is the most common type of lupus. SLE is an autoimmune disease in which the immune system attacks its own tissues, causing widespread inflammation and tissue damage in the affected organs. It can affect the joints, skin, brain, lungs, kidneys, and blood vessels. There is no cure for lupus, but medical interventions and lifestyle changes can help control it.
Next, we will continue with innate immunity. Innate immunity is the first response of the body's immune system to a harmful foreign substance. Key feature for innate immune is cell are non-specific. Innate immunity responds fast, in which happen in minutes to hours. No memory associated with innate response, and will respond to the same pathogen in the exact same way, no matter how many times it sees the pathogen. The innate response include mechanical barrier, for example, skin, mucous membrane, fluid such as tear and urine that physically block pathogen from entering the body, and lastly, the cilia that sweat mucus and pathogen fluid body opening where they can be removed from the body. Next, chemical barrier. Chemical barrier in innate response include enzyme in sweat, saliva, and cement that give pathogen on body surface, and there are also low pH in tamas, kill pathogen that enter the gastrointestinal tract in food or water. And lastly, biological barrier. Biological barriers are living organisms that help protect the body. Millions of harmless bacteria live on the human skin. Many more live in the gastrointestinal tract. The harmless bacteria use up food and space, so harmful bacteria cannot grow. For the next slide, we can see the different type of barrier in innate immunity. Last but not least, let's know about cellular response of the innate immune system. This response involves a variety of different types of leukocytes. Many of these leukocytes circulate in the blood and act like independent, single-cell organisms, searching out and destroying pathogens in the human host. These and other immune cells of the innate system identify pathogens or debris and then help to eliminate them in some way. This is an overview of innate immunity.
Historically, SLE is considered as an adaptive immune system disorder. Adaptive immunity occurs after exposure to an antigen, either from a pathogen or a vaccination. If the innate response is insufficient to control an infection, then adaptive immunity will activate. There are two types of adaptive immunity, which are cell-mediated immune response and humoral immune response. In this, adaptive immunity consists of two major components, which are lymphocytes that consist of T cell and B cell. So in the bone marrow, involve the activation of B cell into the plasma cell, and then the plasma cell will produce antibodies, and then the antibodies will be released to blood and lymphatic fluid to exert its biological function. While in the thymus, immature T cell will mature in the thymus to divide and differentiate to become three subsets of mature T cell, which are T helper cells, cytotoxic cells, and regulatory suppressor T cell. Next is cell-mediated immunity. It is regulated by T cells via direct lysis of target or infected cells by cytotoxic T cell. It is the production of cytokines that activate infected cells to kill pathogens. Last but not least is humoral immunity. So there are two types of activation of B cell, which are T cell independent activation of B cell and T cell dependent activation of B cells. And it is mediated by antibodies, which are produced by activated B cell.
Now, let's see how immune dysfunction may lead to SLE. Firstly, the disease is characterized by the production of self-autoantibodies that is directed against nuclear self-autoantigens, inflammation, and organ damage. Secondly, the presence of anti-nuclear antibodies has been detected in the serum of a majority of patients before the onset of clinical disease symptoms, and levels of certain autoantibodies have been found to correlate with disease activity, supporting a role for these antibodies in mediating disease pathology. Thirdly, it is thought that these antibodies form antibody nuclear antigen immune complexes which deposit in tissues and trigger local inflammation, thereby contributing to tissue injury. Next, increased apoptosis, that is programmed cell death, and defective clearance of apoptotic material are characteristic of human SLE. Autoantigens, typical of lupus cluster in surface, blebs of apoptotic cells, increasing their immune exposure. Moreover, saturation of physiological processes to safely remove apoptotic debris amplifies autoantigen exposure will happen. Neutrophils are the most abundant leukocyte in human blood and one of the first immune subsets to respond to a microbial insult. However, dysregulation in both their function and cell death has been reported in SLE. The increased formation and decreased dismantling of neutrophil extracellular traps (NET) is also thought to be a source of autoantigen self-autoantigen exposure. A granulocyte, gene signature which may relate to the presence of an inflammatory subset of neutrophils called low-density granulocyte has also been shown in JSLE patient.
That's all from us. Thank you.